Hi everyone, and welcome to our third TIA Centre seminar of the academic year. So for those of you that are new to our seminars, my name is Adam Shepherd, and I'm a postdoc here in the TIA Centre. And in these seminars, we aim to invite people from across the globe to present new and exciting work. So today, I would like to introduce Anglin Dent. Anglin is an MD/PhD student at the University of Toronto. She previously pursued an MSc in the laboratory in laboratory medicine and pathobiology under the supervision of Dr. Phidias Diamandis, where she studied the utility of leveraging unsupervised deep learning algorithms to automate the detection of biologically distinct tumor subregions for molecular characterization. The title of Anglin's talk is HAVOC, Small-Scale Histomic Mapping of Cancer Biodiversity Across Large Tissue Distances Using Deep Neural Networks. So Anglin, thank you very much for joining us, and yeah, we look forward to your talk, so please go ahead and...
Great, thank you so much. I think that was a great introduction. So yeah, thank you for having me. Today I'll be presenting work from my master's thesis and that we recently published. And I'll be describing our HAVOC platform, aiming to map cancer biodiversity across tissue distances using unsupervised deep learning methods. Okay. So I like to start off with this relatively simple slide, and I'm sure we're all familiar with intratumoral heterogeneity, but given how many times it's mentioned in this talk, I feel like it's a good introduction to bring us back to what exactly intratumoral heterogeneity is. So in the context of cancer, intratumoral heterogeneity presents a major challenge and wreaks havoc for the treatment of cancer across various malignancy types. So the presence of these spatially distributed clonal diversity within individual tumors has been suggested to be a significant barrier for the success of current therapeutic strategies, as well as a contributor towards disease progression. And unraveling variations in tumoral biology within the individual tumor is not a simple task. So firstly, we need to consider just the sheer number of cells within a tumor and the fact that small biopsies can contain many orders of magnitude higher than what current single cell profiling approaches can process. And in current precision medicine approaches, where tissue is taken from a single malignant location to provide molecular information about the sample, this does not adequately address the need to fully capture tumor biology because we encounter significant sampling bias as well as experience some non-representative undersampling or mixing of critical tumor subpopulations when we perform this type of bulk profiling approach. So our group identified that there's a clear need for a method that is capable of identifying and mapping areas of potential biological variation across large tumor specimens so that we might be able to guide the deployment of limited molecular profiling resources to accurately and comprehensively capture tumor biology. So we considered this problem by borrowing some concepts from geography. So in geography, we have these large scale maps that provide high resolution information of relatively small topographic areas. And they're usually complemented with these smaller scale maps, which are more thematic and document a set of relevant features over larger regions. So small scale thematic maps such as these could also be used to guide systems level decision-making and resource allocation. So we theorize that in the context of tumor heterogeneity, the creation of small scale maps could potentially guide where limited molecular profiling resources can be deployed to better capture the biodiversity of tumors and achieve a more comprehensive understanding of their underlying biology. So we kind of depict our vision for this mapping approach in the bottom here. So building upon this idea of mapping regions of biovariation across individual tumors, our group developed HAVOC, which is a histology-based deep neural network pipeline that aims to create histomic atlases of variation of cancer, with the goal of providing spatially defined estimates of biodiversity across any scale. So this concept and approach was based on two key concepts from both classic and modern histopathology. So the first being that substantial changes in molecular programs are accompanied with modifications in cellular morphology, and that these cytoarchitectural variations can be objectively defined by contemporary computer vision strategies. So I'm just going to share some of our group's previous work to illustrate a couple of these two concepts that I've mentioned. So to illustrate this first concept, I have some figures here demonstrating the concept of divergent biological programming leading to unique downstream histomorphologic footprints. So we highlighted this phenomenon previously in our lab using glioblastoma, which is a notoriously heterogeneous cancer specimen. So here, morphologically distinct niches of GBM, such as cellular tumor, palisading cells around necrosis, microvascular proliferation, and infiltrating tumor regions. They were manually identified, extracted, and then proteomically profiled by our group. And unsupervised clustering highlighted a spatial organization of proteomic heterogeneity in glioblastoma, with these known morphological niches demonstrating distinct molecular profiles. So generally, the application of machine learning computer vision strategies allows for the automation of this morphological mapping process so that it may be applied in clinical scenarios. Prior to the development of HAVOC specifically, our group has illustrated the utility of unsupervised deep learning approaches for completing this task. So here we demonstrated how the application of an unsupervised deep learning pipeline could be leveraged to objectively define regional changes in tumor morphology without explicit instruction. And we showed this by extracting a previously learned feature representations and then applying them to define clinically recognized morphological features and changes in new data sets. So for example, here we've demonstrated how a brain tumor educated convolutional neural network was used in an unsupervised manner to highlight tumor morphologies that included nuclear grade, sarcomatoid features, as well as cystic features. So by applying an unsupervised deep learning pipeline, this approach aims to allow the exploration of distinct histomorphologies without any predefined constraints. So the central question of our recent paper was this: do HAVOC maps of biovariation demonstrate concordance with human-defined and molecularly defined estimates of heterogeneity? So I would now like to take you through exactly how we developed a pipeline that was capable of answering this question, and our results that suggested that, yes, HAVOC is capable of highlighting these properties of intratumoral heterogeneity. So our first goal was to expand on our group's previous findings and assess if the delineation and quantification of spatial transitions across tumors could be automated. So broadly, to assess if spatial transitions in tumor histomorphology could be defined, we applied our previously developed unsupervised deep learning based framework to a cohort of 40 digitized glioma whole slide images. So for example, one right here. And these glioma whole slide images had varying degrees of histomorphologic heterogeneity that could be identified at the human level. So when whole slide images from our cohort were run through our deep learning pipeline, they're first divided into small image patches, non-overlapping image patches, and then subsequently analyzed by our brain tumor histology optimized convolutional neural network. So this convolutional neural network had previously been trained on a diverse set of nearly 1 million pathologist annotated image patches extracted from over 1,000 brain tumor samples. So then rather than carrying out a classification at the final layer as we would in a traditional supervised task, instead we extract the 512-dimensional deep learning feature vector from the penultimate layer of the convolutional neural network. So this DLFV, as we call it, essentially serves to summarize the activation of the individual deep learning features for each tile and create essentially a histomic feature set for each tile of the slide. So these deep learning feature vector signatures are then analyzed by patch level clustering and visualization approaches to spatially map histomorphologic differences and similarities across the whole slide image. So to demonstrate the power of HAVOC to resolve intratumoral histomorphologic heterogeneity, I'm going to highlight this glioma case right here, which had identifiable histomorphologic variations from our cohort of 40, and our assessment of its HAVOC partitioning made the same clinically relevant distinctions that an expert pathologist would. So as I've described, after being run through HAVOC, we were left with these clusterings based on the differences and similarities in the deep learning feature vectors for each tile. And then these 7 different groupings from the clusterings are then mapped back on to the original whole slide image here. And this is done with color tile visualization approaches, which allows us to analyze which components of the of the tumor HAVOC considered to be distinct from one another. So using high power images of the HAVOC defined regions, we can see how this method is capable of defining lesional tissue from normal tissue at a level that an expert pathologist would be able to define. So gray matter versus white matter versus the tumor. But further on the right-hand side of the slide, we can also see that HAVOC has subdivided the tumor areas based on morphologic pattern differences, including areas of infiltrating tumor edge, just cellular tumor, as well as cellular tumor with edema. So further, we've employed confusion matrices, which compare the Pearson correlation of the deep learning feature vectors across the HAVOC-defined regions. And this provides a quantitative approximation of the histomorphologic heterogeneity across the whole slide image. So meaning a higher correlation between the regions would be indicative of low heterogeneity, and a low correlation between the regions is indicative of higher heterogeneity. So for example, in this slide, comparison of normal tissue to the tumor tissue. So for example, the yellow versus the cyan partition had the lowest DLFV correlation and therefore the highest heterogeneity between the two. Additionally, we can generate feature activation maps of salient deep learning features within each HAVOC partition. So this essentially provides some insight into the actual features that are driving this division between lesional tissue and normal tissue, and allows us to actually identify what the morphological features are being recognized by the deep learning feature, and provide some insight at the level that an expert pathologist would be able to define. So next, to assess the generalizability and quantitative nature of HAVOC, we compared HAVOC quantifications of intratumoral heterogeneity, so these Pearson correlation coefficients, to pathologist estimates of variation. So that's what we have here on the right-hand side. So across our cohort of 40 gliomas, we generated these HAVOC-defined color tiled maps and the Pearson coefficient correlation matrices. And we assessed if the deep learning feature vector Pearson correlation coefficient matched what a human estimate of histologic variation would be. So specifically, we defined these human estimates as ranging from severe, moderate to mild. And indeed, we did find, that there was significant difference between the deep learning feature vector correlations of lesional regions that were deemed to be mildly versus severely heterogeneous by expert pathologists.
Can I just ask a quick question here? Sorry.
Yeah, of course.
Do you mind going back to the previous slide? So my question is, so in terms of the violin plot on the right,
yeah.
So the correlation coefficients between, would you sort of expect the mild correlation coefficients to be more similar to the normal brain as opposed to the severe? Or can you explain that a bit to us?
Yes, so we would expect that the normal brain would be the most heterogeneous, because normal to the lesional tissue should have the greatest morphological differences. So that's why we see that the correlation coefficient is the lowest in the tumor versus normal brain there. And then that's the most heterogeneous. And then we go into different categories of heterogeneity, being severe, moderate, mild, severe being the most closely related to this tumor versus normal brain type of heterogeneity. So the least similar tissue and the lowest correlation between the deep learning feature vectors.
Okay, thank you for explaining.
Yeah, no problem. Okay, great. So essentially, this was our first initial question. So it could be concluded from this initial question that HAVOC can provide an automated, objective, and pathologist concordant tool to quantify spatial histomorphologic variation across cancer tissue. So these findings then laid the groundwork to next evaluate if the degree of histomorphologic heterogeneity defined by HAVOC also correlated with underlying molecular differences, which is my next question. So we started answering this question with one of the glioma cases that we found in our original 40 glioma cohort. So here we noted that HAVOC partitioning had captured a BRAF mutated subclone, as indicated here with the IHC staining. And we also identified that tumor subpopulations identified by HAVOC also highlighted distinct Ki67 proliferation indices. So this indicated to us that there was distinct underlying biological programs, both with this mutation that was captured and these distinct proliferative programs that were occurring within the same tumor. So this finding was the first indication that HAVOC identified regional changes in morphology could predict variability in underlying tumor biology. To directly assess if HAVOC's defined histomorphologic heterogeneity correlated with spatial expression profiles, we specifically zoomed in on these cellular tumor niches defined by HAVOC and examined if the HAVOC definitions in cellular tumor regions corresponded with distinct molecular differences identified by global mass spectrometry-based proteomics. So this was assessed in this sample here, which was a primary IDH wild-type glioblastoma, again, a notoriously heterogeneous specimen. So the HAVOC-defined cellular tumor regions that we assessed here were specifically the yellow and the red regions. To profile these regions, the HAVOC-defined regions were used as a guide to complete laser capture microdissection in triplicate, and then as well as a bulk dissection of each sample as well. Once dissected, these samples were assessed at the molecular level with global mass spectrometry-based proteomics to address if the HAVOC-defined regions did indeed predict distinct molecular profiles. So as we can see here, HAVOC did define unique molecular profiles associated with each histomorphologically defined region, so the red being very separate from the yellow. Triplicate bulk dissection of the sample was also included, representing the current method of molecular testing for these samples, and to highlight how distinct profiles are created when we look at the two different regions versus the tumor as a whole. Importantly, the intratumoral molecular differences across the HAVOC-defined regions were also present at the pathway level. So with significant differences in proliferative potential, as demonstrated by an enrichment of MYC programming in the red region, as well as differences in invasiveness, as indicated by an enrichment of KRAS in the yellow region. And finally, as well as in hypoxic programming, which was also greater in our yellow region. So notably, the identification of many of these programs from the red HAVOC region were actually dampened when we profiled the entire tumor specimen. So for example, as indicated by our bulk samples. So for example, here we can see in the MYC enrichment is very visible in the red sample, but then when we profiled the bulk sample, that was not a clear underlying programming that was occurring. So we found that this really highlighted the need for regional profiling of highly heterogeneous tumors when using expression-based techniques. So given the role of MYC in driving proliferative programs, the next step was to validate the findings of this heterogeneous biology with immunohistochemical staining for cell cycle marker Ki67 or MIB1. So to quantify the MIB1 staining index across the HAVOC proposed regions, we used a different convolutional neural network trained in MIB1 scoring, as we can see here in the center. And as we can see, this highlights clear differences between the proliferative potential of the HAVOC-defined regions. So with the red HAVOC region demonstrating significantly higher MIB1 staining than the yellow region here. I've also included some feature activation mapping of the salient features in the proliferative versus the invasive HAVOC-defined regions. So the proliferative being the red up here and the invasive region being the yellow down here. This demonstrated that the activated deep learning features were highlighting patterns of high cellularity and cytoplasmic fibrillary patterns. And this further demonstrated the increased proliferative capacity and invasive qualities of these regions that were driving the HAVOC-defined partitions. And again, this increased cellularity in the red HAVOC partition with proliferative programming was further validated with a MASCAR CNN nuclei quantification tool, and box plots highlighted that there was objectively more cellularity aligning with this increased proliferative capacity in the red versus the yellow HAVOC-defined region. So to see if this observation that HAVOC could identify distinct proliferative programs within individual tumor samples, to see if that held across multiple cases, we then examined if this variability could be predicted by HAVOC using accompanying H&E whole slide images of glioma tumors. So for example, this case here was examined, and we can see that over here on the left, there's distinct tumor histomorphology, some areas with higher cellularity. We can see that when HAVOC resolved this H&E case, we ended up with 7 different regions proposed. And we could see how these HAVOC-defined regions, so the orange, the blue, purple, yellow, and red, predicted tumor regions that had stark differences in proliferation indices, as indicated on this graph here. So this analysis also highlighted HAVOC's capability of mapping geographically separated subclusters that had distinct morphology and that maintained stable cluster-specific Ki-67 proliferation indices across the slide. So the finding that geographically distinct tissue regions could be grouped together suggested that H&E-based DLFV signatures may generalize to multiple spatially separated tumor regions, and could also and indicated that HAVOC could potentially be used to create maps of biological variation across larger specimens. So the findings of this aim of the paper demonstrated that HAVOC estimates of heterogeneity can be used to identify subpopulations with molecular differences across tumoral samples. And this also suggests that HAVOC might provide potentially a cost-effective means of predicting and triaging spatial areas of relevant molecular differences across clinical tissue samples, and laid the groundwork to next examine if HAVOC could construct maps of biovariation of larger specimens spanning multiple histopathological slides. So this was our next research question: can HAVOC map biovariation across centimeter scale distances in larger specimens? So this challenge of mapping large specimens is highlighted here in this case, which is a large recurrent IDH mutated anaplastic oligodendroglioma. This tumor spanned 12 individual slides, A to L, and was 4.4 centimeters in its maximum dimensions. So using this case, we assessed the capability of HAVOC to define reoccurring spatial areas of heterogeneity across all 12 slides. So using all 12 digitized whole slide images from the specimen resection, HAVOC was used to perform image-based clustering and identify 7 regions of interest per H&E section across the whole specimen, and that is summarized here. So in total, we had 84 HAVOC-defined regions. So essentially, differences and similarities of the deep learning feature vector activation of the 7 regions from the 12 slides, so 84 clusters in total, were then arranged using this Pearson correlation matrix. And this identified multiple different reoccurring HAVOC-defined niches. So essentially, when we're looking at this matrix, each slide is represented by a color on the leftmost panel. So as we can see, the deep learning feature vector activation does not cluster according to slides, or else there would not be this intermixing across the slides. We can see that this Pearson correlation assessment indicates that there were roughly 7 patterns of similarities across the 12 slides that were reoccurring and grouping together in this matrix. And those 7 are sort of summarized here. So then when we took a closer look at those 7, we first saw that there was a strong association with distinct histomorphological patterns, including groupings of hypercellular cellular tumor nodules, high cellularity tumor, moderate cellularity tumor, and normal white matter. So the third column in this graph now represents these pathologist-defined patterns of distinct morphology, and we can see how they're clustered together, the nodule corresponding to the pink on this graph, and high cellularity being beige, moderate cellularity being purple, and normal white matter being olive. Further, when we compared the proliferative indices of these reoccurring morphologically distinct HAVOC proposed groupings, we also saw significant differences, with the hypercellular nodule having significantly higher MIB1 staining, and then progressively lowering across the HAVOC-defined niches. And this was, again, estimated by an expert pathologist. So to further expand from the findings that biological programming, so in the previous case, proliferative programming, could be identified across large tumor specimens, we then performed a few experiments to assess the general ability of HAVOC to map regional molecular profiles across larger tumor distances. So here we examined some paired slides from individual IDH wild-type high-grade gliomas. So in these analyses, each slide from the individual sample underwent HAVOC mapping, so to produce these color tiled maps. And then we completed laser capture microdissection and mass spectrometry-based proteomics of these HAVOC-defined regions of the cellular tumor again. So in this first case illustrated here, the regions analyzed were green and yellow from sample 2A, as well as red, yellow, and blue from sample 2B. So we can see here that HAVOC identified a focal hyperdense outlier region, which was 2B blue, that was distinct in terms of its DLFV correlations as well as its proteomic profiles. And the remaining regions across these tumors demonstrated a reciprocal concordance of proteomic profiles that were in line with their DLFV correlations. So specifically, the green region from 2A had a higher DLFV and proteomic correlation with the red region from 2B, while the yellow region from 2A had a higher DLFV and proteomic correlation with the yellow region from the slide 2B as well. So therefore, HAVOC was able to define regions from separate slides that had both a greater DLFV and proteomic correlation than with subregions on the same slide. And again, this is just another sample illustrating that HAVOC, again, was able to partition reciprocally aligned, or that HAVOC partitions were reciprocally aligned with appropriate regions on different slides. So the DLFV correlation of these HAVOC regions aligned with the proteomic profile correlation of those HAVOC regions. So to summarize the findings from this aim, HAVOC was applied at a broader level than that of the individual slide to make clinically and biologically relevant distinctions across larger tissue distances. So given the ability of HAVOC to reveal molecular differences across individual samples, we next wanted to examine if HAVOC-guided profiling could reveal previously unappreciated biological insights. So we theorized that a HAVOC-based multi-region sampling approach could potentially reveal aspects of tumor biology that may not be readily accessible from a standard single-region profiling strategy. So to answer this question, we wanted to assess if HAVOC was capable of identifying patterns of regional variation of cellular states within individual tumors. So to do this, we aggregated the molecular profiles of 19 regions spanning 6 IDH wild-type high-grade gliomas and did an unsupervised clustering analysis of their profiles using a predefined set of 64 proteogenomically concordant molecular profiles. So though we did find that clustering was partially driven by patient ID, so highlighting again this both an intratumoral heterogeneity, but also a high intertumoral heterogeneity between patients, we were able to identify spatial variations in the molecular programs of all these cases, including regional patterns of immune response, hypoxic response, proliferation, and embryonic differentiation states. And when we did a UMAP dimensionality reduction, we saw that there was significant influence of the differentiation signatures on these different programs that were occurring. So specifically, there was an embryonic poorly differentiated cell state and a mature astrocytic phenotype that kind of drove these different programmings. And this was further supported by the fact that these two distinct molecular signatures, this embryonic and the astrocytic phenotype, had a strong inverse correlation here. So when we specifically looked at these two states of differentiation, we found that across this samples that we had, there was significant spatial differences and a high occurrence of these two contrasting patterns co-occurring within individual tumor samples. So this held some clinical significance for us, as tumors are commonly characterized as being either poorly or well differentiated upon clinical presentation. So the fact that when we looked at the programming of specific HAVOC-defined partitions and saw that there was the presence of both poorly and well differentiated tumor subpopulations within a singular tumor, this further highlighted to us the importance of this multi-region sampling approach to achieve a comprehensive understanding of tumor biology and tumor behavior as well. And we sort of further emphasized the importance of these findings by extending this identified astrocytic embryonic axis to the bulk molecular profiles of glioma cohorts within the Cancer Genome Atlas. And we demonstrated that this either embryonic or astrocytic state had previously been correlated with tumor aggressiveness, consisting of either tumor grade or the presence of an IDH mutation. So these findings of differentiation state correlation with tumor aggressiveness also just further emphasized to us the importance of profiling multiple tumor subpopulations with distinct differentiation profiles. So to recap that aim, we identified that HAVOC is capable of revealing previously unidentified patterns of spatially organized molecular heterogeneity. And then the last aim of our overall project was to examine the generalizability of HAVOC. And specifically, we wanted to examine if it was possible to generalize HAVOC to non-CNS neoplasms, as well as alternative molecular profiling differences. So we first did this using a publicly available H&E stained section of an experimental metastatic lung cancer mouse model. So this came from a paper originally published in 2021. And in this paper, 2 independent clones of a mouse lung tumor were injected into the tail vein of a mouse to result in multiple spatially distinct liver metastases. 2 of these metastases, as seen in this red box here, were then characterized in the original study using spatial DNA profiling technology and RNA sequencing. And they were characterized as being both genomically and transcriptomically distinct. So when we looked at this tumor section with HAVOC, we originally generated 11 partitions, as demonstrated here. And those 11 partitions segmented the 5 tumors into 2 major classes, in addition to defining some regional histological patterns of liver tissue that were also characterized in the original study. So HAVOC was able to delineate the regions that were previously defined in the original study as genomically distinct, so specifically the green versus the blue region here, as well as I mentioned, identify the peritumoral liver that had inflammatory cells present in it as well. So importantly, when we were looking at the feature activation mapping of these different HAVOC partitions, we saw that there were distinct clinically recognized morphological features in the HAVOC-defined regions. So specifically in clone A, our green region, we could see that the features were identifying advanced organization of tumor cells with nuclear palisades. And in the blue region, we could note that the features were identifying a more random arrangement of large atypical cells scattered throughout the lesion. Additionally, I just want to mention that within the peritumoral liver tissue, feature activation mapping identified inflammatory cells as well, and that was in line with the original tumor, normal, and immune cell classes that were assigned from the original study. So we can see down here that specific immune cells were being identified by that feature. So next, using our own specimens, we wanted to examine the generalizability of HAVOC to different tissue types. So we first started by examining this dermatopathological specimen that had been characterized as a neuroendocrine collision tumor. So this tumor had distinct CK34, BE12, and synaptophysin immunohistochemical staining, highlighting the distinct squamous and neuroendocrine portions of this tumor. So as we can see in the bottom left here, HAVOC appropriately distinguished this distinct squamous and neuroendocrine morphologies in line with the immunohistochemical staining ground truths. So specifically, the green cluster here from HAVOC matched the squamous morphology, and the blue matched the neuroendocrine morphology of this tumor. Additionally, in this pulmonary specimen, we saw that there was divergent adenosquamous tumor differentiation with distinct TTF1 and P40 staining. So HAVOC partitioning of this case demonstrated a high spatial concordance to TTF1, so the adenocarcinoma portion, and the P40, the squamous carcinoma tumoral component. Additionally, when we looked at the hierarchical clustering of the HAVOC-defined regions, we saw that there were two distinct tumor sub patterns, and that DLFE correlation coefficients demonstrated that the divergent P40 positive staining pattern, so this one here, had the lowest DLFE correlation value among the tumor regions. So to summarize our findings from the same, HAVOC biodiversity maps generalized to untrained tissue types as well as genomic differences. So at this point, I'd just like to recap what we found. So we found that HAVOC is capable of systematically and quantifiably mapping histomorphologic variations of cancer with a strong concordance to human-defined and molecularly defined estimates of heterogeneity. We also found that it's capable of mapping tumor histomorphological and molecular variations across large tissue distances, such as entire tumor specimens. And we also found that it's capable of generalizing to other tumor types and cancer models. When we looked at what HAVOC is capable of revealing, we saw that tumor subpopulations with varying degrees of phenotypic differentiation and proliferative capacity can coexist and be geographically separated. And this further underscored the need for more intelligent sampling strategies to us. Which brings me to the clinical significance of our findings. So we theorize that the routine generation of HAVOC mapping of biovariation in cancers has the potential to identify and document critically divergent cellular habitats across large tumor regions. So this has the potential to help manage tumor heterogeneity, both for large cohort studies, but also personalized medicine efforts by guiding the deployment of limited profiling resources to the most relevant and biodiverse tumor niches. So I'd like to specifically just discuss the benefits of HAVOC, since there are so many different profiling methods available, as well as we know, computer vision approaches aiming to target these problems. So HAVOC specifically aims to address this disconnect between the limitation of tissue profiling capacity and tumor sizes. And it's been designed with the idea of complementing alternative computational approaches that are able to quantify a very specific features in a supervised manner by mapping intratumoral heterogeneity in this hypothesis-agnostic manner. So specifically, HAVOC employs a hypothesis-free approach to identify at-risk regions as spatial targets for prospective and personalized profiling. So this will not rely on any set of predefined features that may bias or limit the exploration of biological variation. However, we do identify that this does potentially compromise the sensitivity for detecting subtle spatial variations, but it does allow, this is sort of the trade-off that we have for providing a highly dynamic and generalizable approach for personalized discovery and also characterization of spatial changes in tumor biology. Also, just some sort of smaller details that are quite beneficial in terms of actual processing of data. The HAVOC method does not require any dedicated tissue or computational resources. It requires only that you have a whole slide image of your sample and can be uploaded to our cloud-based server. Additionally, there are minimal tunable parameters to our approach. So tile size and cluster number can be adjusted depending on the complexity of tissue, but essentially the processing and completion of these HAVOC maps are fairly straightforward and user-friendly as well. But of course, I cannot talk about the benefits without discussing some of the limitations, and inherently the dependence on tissue pattern does introduce a vulnerability to artifacts, and they are readily identifiable by HAVOC, and those include tissue folds, tears, and focus errors. However, we have found that in our analysis, these are readily apparent and can easily be excluded from analysis, whether that be post hoc or doing a type of supervised clustering to remove that class from the HAVOC partitions. Additionally, kind of feeding back into the tunable parameter sense, the sensitivity for very subtle and intermixed patterns does need to be assessed in a context-specific manner to identify what molecular profiling technique is best suited for that analysis, but also if sort of tile size and number of partitions needs to be changed as well. And then lastly, some additional future directions beyond simply like the clinical significance of these findings. Our group has also been using HAVOC in computational pathology research as well, and is encouraging people to use it as well as a way of developing and training custom classifiers. So essentially what we do is we run the slides through HAVOC, generate these sub-classifications, and then it allows the user to label each classification to their own specification and then generate a classifier based on the tiles generated from those HAVOC partitions. So that's just another exciting application of this tool too. And with that, I would like to acknowledge everybody from the Diamandis Lab specifically that helped with this project, and as well our funding sources. And thank you so much for listening. I'm open to any questions, of course.
Brilliant. Well, thank you very much, Anglin, for a really great talk. And yeah, with that, we'll open the floor to questions. So does anyone in the room or online have any questions they'd like to start off with? In that case, I'll start then. So in terms of actually using HAVOC as a tool to help a clinician, for instance, how do you envisage doing that? Is it a case of literally providing these HAVOC maps potentially without labels, and they'd be able to spot these particular regions of interest that are showing heterogeneity? Is that the
Exactly. I think you have it exactly. So what we envision is having, essentially, you have the tumor extracted, you have your whole slide image, and before any molecular profiling approaches are deployed, we would envision that you run it through HAVOC, examine the cellular tumor regions that are identified, and sort of provide like a more intelligent approach for determining what regions of the tissue should be sampled to achieve that more comprehensive understanding.
And a sort of follow-up on that, I suppose, is so you said that you see issues when it comes to various different artifacts such as folding, but have you seen, do your features change much when you look across domains? So for slides that have been scanned on different scanners or from different centers, what's the sort of the robustness or the variability that you get as a result of that?
Yes, so we do have specifications for HAVOC. And that being that all of our scans are scanned at, I believe it's, let me double check exactly what it is. Yes, so they're scanned with a compression quality of 0.7 and a magnification of 20. So the scans do need to have that, basically those qualities for HAVOC to work, and we definitely did notice when we looked at scans that were maybe scanned at a different compression quality that we were seeing some artifacts, because you can imagine that if it's magnified to a different degree, that the nuclei suddenly don't look like nuclei anymore. And so all of our training slides were trained with those parameters, and then our the slides that we've assessed are at that parameter as well. So that definitely is within our center, that is the standard for the slide scanning. But at some limitation, I guess, to sort of wider deployment would be if other centers were using different parameters for their slide scanning. So that is another potential limitation. And at the moment, we have it sort of as a caveat on our cloud-based server that those are the only types of slides that we can process.
In terms of staining variation, so if there was any differences in the actual protocols to stain and to section the slides, etc., have you seen any effects of that at all? Or
we currently have. Sorry, could you?
No, continue, please. Sorry.
Yeah, so we currently have not seen any issues with that so far. If I'm understanding your question correctly, given that is the standard scanning like protocol for our clinical center as well, and we were using clinical specimens, that has so far not been an issue, but I think as we expand, that might come up as a limitation or something that needs to be adjusted.
Brilliant. And I suppose as a lot of computer scientists in this room probably thinking the same sort of questions in terms of, so the backbone for this HAVOC model is a VGG, right?
Yes.
And it's been trained on, it's a private data set, I'm assuming, right?
So let me bring this up. So we had our VGG 19. We did do transfer learning to optimize it on neuropathology image patches to recognize specifically neuropathology patterns, although we did identify in some previous studies that essentially, given that many morphological features are shared across cancer types, that we were able to leverage these previously defined features, I guess, as I showed today as well, to cancer types beyond just brain specimens as well.
And is this pre-trained network available, or?
Yes, so this is the pre-trained was sort of like a out-of-the-box VGG 19, and then we had our trained by transfer learning. That's the model that's available on our cloud server.
Okay, yeah. Oh, brilliant. And did you by any chance manage to compare it, so the outputs of your of HAVOC, I suppose, did you manage to compare it to other sort of pre-trained networks in pathology, such as like CTransPath or anything like that?
We did not. This was our essentially what we started with and have done our previous studies with and then grown from here. We did have a few comments from reviewers that we addressed where we compared, I'm not sure if this is exactly what you're asking, but we compared sort of our transfer learning neuropathology optimized VGG to the just sort of out of the box VGG. And we did see that there were, that essentially when we had it trained on the transfer learning, that it was able to achieve these more intelligent and sort of pathology optimized distinctions much faster than when it was not trained with transfer learning.
Oh, very cool.
Mm-hmm.
And so do we have any more questions for anyone online or in the room? Yeah. Do you want to, can you?
Have they tried this on other cancer types? This is trained on brain.
Yeah. So the question from the question was, have you tried to use this tool on other cancer types? So aside from brain, have you tried it on breast cancer or?
Yes. So the last couple of slides that I demonstrated here, sorry, I'll just scroll back. So we demonstrated here on a couple skin cancer and lung cancer types that we had some very similar findings as well. So in terms of demonstrating that it was capable of delineating sort of biological distinctions across different tumor types, we were able to do that in the study as well. In previous studies, we also showed, we have a really cool JCO paper that kind of laid the groundwork for using this unsupervised pipeline across all the cancer types in TCGA. So we demonstrated that we could define these distinct morphologies across tumors that spanned every region in the body. So that was a really cool finding.
Brilliant. Well, if we've got any more questions online or in person,
I guess
have you?
one more.
Of course.
So in terms of the pipeline for this whole thing, if for example, you wanted to plug and play the kind of a more general feature extractor such as C-transpath instead of the brain specific one, and then kind of plug that model into the HAVOC pipeline, if that makes sense. How hard would that be to do, and do you think it would make sense to do it?
Yeah. So I have to provide the caveat that I am not the computer scientist that designed our pipeline. So that is probably a better question for our my co-author on this paper, who was our in-house computer scientist for this. So that's definitely a good question, and something that I could get back to you on if once discussing with him.
Cool, thank you.
Thank you.
Fantastic. Well, with that then, I suppose we'll call into the seminar, but thank you so much for a really interesting talk. And I really liked I looked at the website that you've got on your paper as well. And it's a really cool tool. So nice work.
Great.
But thank you very much for joining us.
Thanks for having me. Thank you.
And just a reminder for everyone online and in person, we've got our next seminar is in 3 weeks' time on Monday, the 27th of November from 2 to 3, where we'll be joined by Dr. Pingjun Chen from the University of Texas. So hope to see everyone there, and thanks again, everyone, for all the questions, and thank you again, Anglin, for a fantastic talk. See everyone next time.